Today, we'll be doing a mind map on cellular membranes and seeing the applications of it in respiration. Respiration occurs in mitochondria whose structure can be seen here. However, before we go into that, we first have to look at membrane structure. The first model to describe membrane structure was proposed by Davison and Danieli in the 1930s. Their model, sometimes referred to as the sandwich model, was developed from observing electron micrographs, which seemingly depicted membranes as a lipid layer sandwiched between two protein layers. However, in the 1950s and 60s, new evidence in the form of freeze-fracture electron micrographs, analysis of polar properties of membrane proteins, and experiments using fluorescent markers falsified the Davison-Danieli model. A new model was thus proposed by Singer and Nicholson in 1966, referred to as the fluid mosaic model. You can look more into the history of cell membrane theory as this can come out as a nature of science inquiry. What you see here is a basic depiction of the fluid mosaic model, which is the currently accepted model of membrane structure. As discussed in my mind map on cell structure, cell membranes act to separate cytoplasm, which is everything within the cell, from extracellular fluid, which is everything outside the cell. The cell membrane is made up of phospholipids, which naturally adopts this bilayer arrangement. The fatty acid tails point inward towards each other, while the phosphate head points outwards. This is due to the amphipathic nature of phospholipids. What this means is that part of the phospholipid molecule is attracted to water, while part of it repels water. The fatty acid tails, which are hydrophobic, repel water and are instead attracted to each other. On the other hand, phosphate heads, which are hydrophilic, are attracted to water. Therefore, this is why the phospholipid membrane forms the bilayer. As you can see, the Singer-Nicholson fluid mosaic model shows membrane proteins embedded within the phospholipid bilayer and not just layering the outer surface. These proteins are called integral and peripheral proteins respectively. There are many different types of membrane proteins, one example being channel proteins with multiple functions. These include hormone binding sites, where an application of this being the regulation of blood glucose levels by insulin. Secondly, enzymatic reactions, where immobile enzymes have their active sites sticking out into extracellular fluid. Also, there is cell-to-cell adhesion and cell-to-cell communication, where an application of this being the receptors at the synapses of nerve cells. Lastly, we have channels for facilitated diffusion, as well as pumps and carriers for active transport. Do note that these membrane proteins are not fixed in one position, but move fluidly within the phospholipid bilayer. The last two examples are related to transport of substances across the membrane. Substances move across the membrane via four means of transport. Firstly, simple diffusion, where particles simply pass through the bilayer in between phospholipids along the concentration gradient. This is usually for smaller and non-charged particles. Secondly, osmosis, which is for water molecules. Thirdly, facilitated diffusion. This occurs through channel proteins where particles that cannot move across the membrane via simple diffusion pass through channels instead down the concentration gradient. The structural and chemical properties of the channels are such that they are also often specific to one substance. Cells control channel production, which allows it to control what substances are taken in. We will see an example of facilitated diffusion later on in respiration. Lastly, we have active transport. This is carried out by pump and carrier proteins, which transport substances across the membrane against the concentration gradient using ATP. An application of this is in the propagation of nerve impulses in neurons. Glycoproteins here help stabilize the membrane structure, while cholesterol controls membrane permeability. Cholesterol controls membrane permeability by maintaining just the right level of fluidity. If the membrane is too fluid, it cannot control what passes across it. On the other hand, if the membrane is not fluid enough, movement of substances within it and across the membrane is restricted. With cholesterol, it disrupts the regular packing of phospholipids and allows for molecular motion. This prevents crystallization and the membrane behaving like a solid layer. The right amount of cholesterol basically allows for the right amount of molecular motion so that the membrane neither acts too much like a solid or a liquid, to best regulate membrane permeability. Sometimes, substances are transported in vesicles, which is a structure containing liquid or cytoplasm, enclosed by a phospholipid bilayer. It is used in transporting substances from one side of the membrane to the other, through either endocytosis or exocytosis. In endocytosis, the membrane is pulled inwards in the direction of where the vesicle will be formed, before pinching off from the plasma membrane. Exocytosis is basically the opposite of this. This ability for vesicles to be pinched off from and fused with plasma membrane is due to its fluid nature and amphipathic properties. Vesicles serve a very important role in cellular activities, which is discussed in my mind map on cell structure. Now on to respiration. Respiration occurs in mitochondria, which is seen here. It is a process that occurs in living organisms to produce energy in the form of ATP. Mitochondria have two membranes, the inner and outer membrane. The reason for this is the folding of the inner membrane to form cristae at the intermembrane space. The space within the inner membrane is filled with a substance called matrix. The formula of respiration is as follows. This formula will be broken down as I go into the 5 steps of respiration. Respiration begins with glycolysis, which occurs in the cytoplasm of cells. One 6-carbon glucose molecule goes through 2 rounds of phosphorylation to form fructose biphosphate. This reaction is catalyzed by hexokinase enzyme, which uses 2 molecules of ATP. Subsequently, fructose biphosphate undergoes lysis to produce 2 molecules of triose phosphate. These 2 molecules then go through an oxidation process to become 3-carbon pyruvate molecules. This oxidation reduces a NAD molecule to reduced NAD, where reduced NAD is now an electron carrier. The role of reduced NAD will be seen in the electron transport chain later on. The oxidation of triose phosphate to pyruvate also converts 2 molecules of ADP to ATP per pyruvate molecule, totaling 4 ATP molecules. Considering that the first reaction of phosphorylation used up 2 molecules of ATP, there is a net yield of 2 ATP molecules per glucose molecule that undergoes glycolysis. This net production of ATP is an instance of substrate-level phosphorylation. Once glycolysis occurs in the cytoplasm, pyruvate molecules move into the matrix of mitochondria to undergo the link reaction. Coenzyme A first binds to pyruvate, where in the presence of oxygen, it undergoes decarboxylation to form acetyl coenzyme A and carbon dioxide. This process of decarboxylation also reduces 1 molecule of NAD to reduce NAD. As glycolysis of one 6-carbon glucose molecule produces 2 3-carbon pyruvate molecules, this link reaction occurs twice to produce a net yield of 2 acetyl coenzyme A molecules per glucose molecule. The next step of respiration is the Kreb cycle, which occurs in the matrix as well. The Kreb cycle begins with acetyl coenzyme A and a 4-carbon compound called oxaloacetate. The acetyl group of acetyl coenzyme A is transferred to this compound to form a 6-carbon compound called citrate. Citrate goes through 2 rounds of oxidative decarboxylation to form a 4-carbon compound. Each round of decarboxylation reduces a molecule of NAD to reduce NAD. A molecule of carbon dioxide is produced as well. This 4-carbon compound then undergoes rearrangement to reform oxaloacetate. This rearrangement reduces a molecule of NAD and FAD to reduce NAD and reduce FAD respectively. The rearrangement also converts a molecule of GDP to GTP, which is another energy-carrying molecule similar to ATP. Therefore, the rearrangement process in the Kreb cycle produces energy, and just like in glycolysis, is an instance of substrate-level phosphorylation. As 1 molecule of glucose feeds 2 acetyl coenzyme A molecules to the Kreb cycle, this cycle repeats again to produce a net yield of 2 GTP, 6 reduced NAD, and 2 reduced FAD molecules. Reduced NAD and reduced FAD are both electron carriers, which is important for the next step of respiration. One fun thing to note is that if you are to total the number of CO2 molecules produced thus far, it will sum up to 6 molecules for 1 molecule of glucose, explaining the formula. The next step of respiration is the electron transport chain, where we see an application of what we have learned about membranes. The electron transport chain occurs at the inner mitochondrial membrane, which separates the matrix from the intermembrane space. As seen, the electron transport chain involves several proteins, which we will go into one by one. In the previous steps, one byproduct that kept being produced was reduced NAD and reduced FAD. As mentioned, both act as electron carriers. At protein complex 1, reduced NAD is oxidized to NAD. As a result of this reaction, 2 electrons are lost to complex 1, which acts as the first carrier of the electron transport chain. The electrons then move along the chain to coenzyme Q. The oxidation of reduced NAD also produces protons, where complex 1 acts as a proton pump to transport protons into the intermembrane space. At protein complex 2, reduced FAD is oxidized to FAD. Just like at complex 1, electrons are lost to complex 2, which joins the electron transport chain as it moves along to coenzyme Q. Complex 2 differs to complex 1 in that it does not act as a proton pump to transport protons to the intermembrane space. After coenzyme Q, the next carrier in the chain is protein complex 3. Complex 3 acts as a path for the electron transport chain as well as a proton pump. Electrons then move to cytochrome C protein, which transfers it to the last carrier of the electron transport chain. Electrons are lost to oxygen held at protein complex 4, which reduces it. Reduced oxygen then picks up hydrogen ions in the matrix to form water. Complex 4 additionally acts as a proton pump. Of the 4 complexes involved in the electron transport chain, 3 of them act as proton pumps. The reason for pumping protons into the intermembrane space is to build up the concentration of protons in the space, to establish a steep proton gradient between the intermembrane space and the matrix. This is crucial for chemiosmosis to occur, which is what produces energy in the form of ATP. There are 3 features which aid the establishment of this steep proton gradient. Firstly, the large number of proton pumps in the inner mitochondrial membrane, which we have already seen. Secondly, the intermembrane space is really small, which allows the concentration of protons to build up quickly. Lastly, the formation of water that occurs at protein complex 4 reduces the number of protons in the matrix, allowing for a steeper gradient. The maintenance of a steep proton gradient is for the purposes of diffusion of protons back into the matrix. This occurs via facilitated diffusion, where protons pass through ATP synthase protein to re-enter the matrix. The force of protons diffusing through this channel protein activates the molecular motors of ATP synthase, which facilitates the phosphorylation of ADP to ATP. This process is referred to as chemiosmosis. For every 2 protons that passes through ATP synthase, 1 molecule of ADP is converted to ATP. Therefore, energy is produced, stored in the form of ATP. As long as the proton gradient is maintained, chemiosmosis can continuously occur to produce energy. However, this is only possible if the previous 4 steps can occur, which is only possible in the presence of sugar and oxygen. This brings me to the end of my mind map on membranes and respiration. Hope you find it useful and have a good day.